Okay, so welcome to day 3 of the Introduction to Single-Cell RNA Sequencing workshop. Thank you for staying with me this far. Recently, I think yesterday, the Sahita lab, who is a leading lab in the development of single-cell RNA-Seq analysis techniques, published this comic. So hopefully we can enjoy our collective struggles together and have a little bit of a laugh as we work through this stuff. So today is the final day of the workshop, and we'll cover the final steps of single-cell RNA sequencing analysis, which are cell type clustering and the quantitative comparison of transcriptomes. I'll do a quick recap of the concepts we've covered so far, and then we'll discuss algorithms which partition cells into transcriptionally similar groups. Specifically, we'll discuss 3 common clustering algorithms, including hierarchical clustering, graph-based clustering, and k-means clustering. And then in the second part of the lecture, we'll go over methods for the downstream analysis of single-cell RNA-seq data, including differential expression analysis and pseudotime algorithms. And in case you were wondering, these cells are cells that we've been protecting for the last year. They're different types of cells lining the bronchiole of our respiratory tracts. So far, we've covered a number of experimental approaches to single-cell RNA sequencing and many of the steps in the standard analysis workflow that aim to prepare your cell data for cell type clustering. So we've discussed how to use quality control filters to remove low quality cells and what effects normalization can have on your data. We've also talked about how to perform feature selection to distinguish between technically varying and biologically varying genes. And we've discussed how to use dimensionality reduction tools like PCA to further reduce the dimensionality of the data, and how to use manifold learning algorithms like t-SNE and UMAP to visualize high dimensional data in 2D space. So today the focus will be on how to cluster cells based on their calculated distance matrices, and then how to quantify transcriptional differences between these groups. Yeah, so hold on. I will share with you the course material for today in case you wanted to, one, download the course material beforehand. Before the demo, there's a bit more material to cover or to download onto your computer today. So that is here. And then the slides. Does anyone have the link that Eloy sent out open on their computer with the slides? Thank you. Okay. Okay, let's start the share again. Okay, today we're going to talk about cell type clustering and the quantitative comparison of different groups. So this is the final goal of many single-cell RNA sequencing studies. Like I've said, all of the prior steps serve to create the cell-by-cell distance matrix, which aims to best represent what we think are the major factors that contribute to differences between cell types. So feature selection is motivated both by the curse of dimensionality and the idea that we can model technical variation to identify a basis of biological varying genes. And the biological varying genes somehow combine to give rise to different expressions of cellular identities. And we use unsupervised clustering approaches to try to find groups of cells that vary in similar ways. So once we have these groups, we can quantify how their transcriptomes differ and use these gene distributions as inputs for further computational or wet lab experiments. Okay. So identifying these cell clusters critically determines the output of your single-cell RNA sequencing experiment. These groups of cells serve as the basis for things like compositional analysis of how proportions of cell types change under different experimental conditions. They also serve as the input for trajectory analysis or pseudotime analysis, which aims to reconstruct cell fate transitions over developmental time. And cell clusters also create the different test groups that are used to compare the relative changes in different gene expression. So the set of genes expressed in each cell cluster are the input for gene set analysis as well, and also inference about gene regulatory networks. And cell clustering is a very important step in the analysis, but it's also mutable. So we've done a lot of work to acquire single cell RNA-seq data, and the clustering analysis can always be revisited and recalculated as improvements in the analytical workflow continue to develop. So while performing cell type clustering, I think it's important to consider the question of what constitutes a cellular identity. The terminology used in the field is cell identity rather than a cell type, as identities are perhaps more flexible things and provisional in single cell RNA-seq analysis compared to the more fixed notion of physiological cell types. And remember, many contexts shape cellular identities and incite the expression of different genes. So for example, different environmental contexts, such as variation in nutrients or hormones, can simulate different homeostatic processes. Cells can also go through changes in their physiological functions as they change compositions during differentiation from a stem cell to a terminally differentiated cell. Cells also change which genes are expressed depending on whether they are proliferating or in a state of quiescence. Also, spatial positions such as distance to blood vessels can change what kind of homeostatic processes are present in individual cells. And I like to think about cell type as something that's analogous to the demographics of people, where identities are kind of dynamically formed in a co-evolution within an environment. And different categories of cells are useful to understand in different contexts, but cells are also multifaceted. So different aspects of their identities might combine in non-additive ways to create something totally different. And identities might not be easily contained by these like static representation of cell types. That's to say, there's a lot we don't know about single cell biology, and what we do know is that we observe a huge amount of heterogeneity at every level of organization. And yet somehow groups of cells interact with each other to maintain different organizational levels of biological integrity. And that's kind of what single cell RNA-seq is trying to figure out. How does this happen? So while I'm not sure about how useful the current concept of a cell type is for our understanding of biology, there are many ways of thinking about cell identity and cell state mathematically. Biology is a very abstract science. So one way of framing cell identity variation is by hypothesizing that different physiological processes may give rise to different manifolds of variation in gene expression space. And this graph illustrates different physiologies as different axes or vectors of variation. But the main point I'm trying to convey here is that when we're studying complex tissues, cells seem to have continuous phenotypes. And we don't actually know whether discrete cell types exist and whether these cell types map to distinct regions in gene expression space. What many single cell RNA-seq studies have shown instead is that we observe continuous variation across and within cell types in gene expression space. And we haven't developed a great framework yet for characterizing this kind of variation. So a goal of single cell RNA-seq studies is to map the boundaries of cell identity in gene expression space. In other words, single cell RNA-seq studies aim to resolve how different physiologies correspond to regions in gene expression space. However, whether or not these distinct cell types or physiologies lie in well-separated regions, it remains unclear. So despite this, the core assumption of current cell type clustering approaches for single-cell RNA-seq analysis is that there exists well-separable regions in gene expression space, or geometries, that map to discrete cell identities, or at least this is the best that we currently know how to do, and we hope that we will learn informative and interesting things. So the standard single-cell RNA-seq analysis procedure recommends combining prior biological knowledge of marker genes with clustering algorithms to try and partition mystery cells into identified cell populations based on their similarities in gene expression. Okay. So what does the theist lab mean when they say combine prior biological knowledge of marker genes with clustering algorithms? Well, it can be as simple as just having a list of several genes that have been previously demonstrated to correspond to important cell physiologies, and using this list of marker genes to annotate your cell clusters. Or the approach can be more sophisticated. Like, for example, people are starting to use machine learning to try to identify the most informative subset of genes for classifying cells. But my words of caution here are the following. Science is an iterative process, and prior biological knowledge is an important reference point, and having new technologies with increased resolution can either provide supporting evidence of prior knowledge, or it can falsify the knowledge and create epistemological turbulence and like sites for new concepts to be created. So we don't want to lean too heavily on prior knowledge to inform our cell clusters, because this prior knowledge created with less accurate or more poorly resolved or a lower number of measurements in the technology, these kind of tools could be misleading to our concept of a cell type. So using this kind of lower resolution knowledge to shape our cell clusters and single cell data can potentially deeply bias the results. But it still is informative to the progress of science. So how do we strike a balance? In some well-annotated model systems, people are working to develop methods that quantify how well clusters fall in line with previous knowledge of the system under study. So in this study of root development in the plant Arabidopsis, the authors used fluorescent microscopy data that mapped the expression of transcription factors and other marker genes to specific tissues within the plant's root system. And Arabidopsis is a really good system to study the development because the maturing cells are spatially organized. So we have new cells generated at the meristem tip. And as the cells transition away from the tip and up the plant stem cell, they differentiate into cell types with different physiologies. So the authors combined previous data to find a set of defined marker genes, and then explored how their single-cell RNA-seq clusters corresponded with the orthogonally generated knowledge of marker gene expression. So we have the marker genes on the x-axis here, and the cell type clusters on the y-axis here. And they also are able to plot 3 developmental categories within their cell type clusters. So you can see how the expression of marker gene changes across cell type and over developmental time. And you also notice that marker genes have stronger expression within these boxes, as is expected, but marker gene expression does not always fall like neatly into the discrete cell categories. It's kind of more the exception that's what we observe. So in this example, they use these sets of genes generated from previous microscopy studies to score the composition of cell types across their experimental conditions. So here they're just plotting the relative abundance of different cell types in mutant conditions relative to wild type, so that the authors can measure how cell type abundance changes as a function of the expression of these interesting transcription factors. And then by evaluating the expression of statistically relevant markers in specific cell types, they're able to predict how the abundance of different cell types change as a function of developmental time, both in wild type and mutant plants, to get a better picture of the meristem development. So one question you might have is, can we use marker genes to do a better job at clustering cell types? And yes, there are a number of clustering algorithms that prioritize cell clustering partitions where the average expression of marker gene expression aligns with the cell clusters. So they're trying to align the expectation with the results of the unsupervised, so it's a more supervised approach to cell type clustering. And in this paper, they don't do that. They're just using microscopy atlas data to annotate which cell clusters correspond to their cell type. But a recent and developing area of methods for single cell RNA-Seq clustering has focused on using these machine learning tools. And these tools aim to identify which set of genes have the strongest classification potential. So marker genes are similar in concept to the goal of quantifying differentially expressed genes. However, in the context of machine learning and cell type clustering, there's a key difference. Differential gene expression evaluates each gene for its expression level variation between groups of cells, whereas marker genes are tested for their classification power. So further, differential gene expression tests each gene's expression individually across groups, while marker genes are tested using machine learning for their combined ability to classify a set of cell types. So there's essentially two main steps in using machine learning to define a set of maximally informative genes for cell type clustering that I'm illustrating here. The first is defining the supervised approach. So this requires that researchers label cell identities prior to training the algorithm. This can be done using orthogonal measures of cell identity, as in this case, they again use this Arabidopsis root fluorescent microscopy data to label cells into classes. But obviously you have to be careful here because how you label your cell types will define your results. And the second step is training the supervised algorithm to identify the set of maximally informative genes for the classification of cells according to the input labels. So here we are finding the set of genes that can be used to best separate predefined cell types. And this is a good way to evaluate how previous notions of cell identity map to single cell transcriptome data. However, having a rigorous set of known cell type markers beforehand is not always the reality. So many researchers are forced to return to the question of how do we perform completely unsupervised clustering of transcriptionally similar cells? Okay. So returning to this question returns us to the cursive dimensionality. In this experiment, authors sequenced a developing embryo in the zygote 2-cell, 4-cell, 8-cell, 16-cell, and blastocyst stage. Here, each cell is projected onto the first two principal components and colored according to the true developmental stage that the cell was in. So when the full set of 20,000 genes was used as the basis for principal component analysis, you see an almost continuous trajectory of the position of cells in gene expression space when cells are plotted on the first two PCs. So here, where the dimensionality is very high, there is not a lot of difference in distance between the groups. And as you can see, there are these zones of indiscernibility as well, where it's difficult and potentially not possible to separate distinct regions of gene expression that correspond to these cells at specific developmental stages. So for example, cells labeled here as at the 2-cell stage overlap with the region occupied by the zygote stage cluster. And the 4, 8, and 16 cell stage form this continuous trajectory over here. But in contrast, when we project the data using only 500 of the most highly varying genes, we observe greater distances between the distinct groups of cells that correspond to different developmental stages. And this illustrates the idea that movement through gene expression space might occur with the continuum of changes, or it might be noise, but narrowing down the subset of genes can narrow down the region in which gene expression can vary and thus increase the space between biologically distinct groups. Okay. So in addition to the curse of dimensionality, people are often concerned about clustering on the original data because highly expressed genes can dominate the distance calculations, just because bigger UMI counts lead to bigger distances in the distance matrix. So often clustering on feature selected data or PCA reduced data can be more robust and give clusters that align more with existing biological knowledge. And while some people cluster cells using t-SNE or UMAP coordinates, don't do this. This will introduce a lot of bias into your data from the topological distortion we talked about yesterday that's produced by these algorithms. So once we've settled on a cell-cell distance measure, there are two general approaches to clustering. One approach clusters cells directly based on their position in this distance matrix, while the other approach looks for the more densely connected neighborhoods of cells. So many different clustering algorithms exist, and today we're going to talk about the three most widely used methods, which include hierarchical clustering, graph-based clustering, and k-means clustering. So first, let's talk in detail about how hierarchical clustering works. In this example, we're plotting pizza places in San Francisco city blocks, and we want to cluster them to find sets of pizza places that are near each other. So the first step in hierarchical clustering is to calculate the pairwise distances between points, and then we group together the pairs that are closest to each other. So we'd start with these two. And this is an iterative process. So we first group the closest pairs, and then we go on to the next closest pairs. And then we come to a point where the next pizza place already belongs to a pair. So when this happens, we simply merge the group to now include the third. And we continue this process. But then we get to a point where the next closest pair already belongs to their own group. And what we do here is simply merge the group. So this process stops when the distance exceeds a certain threshold. So how do we decide what this distance threshold is and how many clusters we want to end up with using a computational approach? So if we plot the pairwise distances, starting with the closest and adding the distances between groups as we merge groups, we simply draw a line at some predetermined distance. And in single-cell RNA-Seq data, it is a standard practice to adjust the number of clusters to match your expectations. The downside of this practice is that using prior expectations can bias your results in a way that misses potentially new or interesting biology, as the number of clusters you choose will alter the resolution of your study of gene expression variation. However, an upside of hierarchical clustering is that it retains information about how subclusters of clusters are related to each other. And this subcluster information can be potentially useful for studying within cell type variation. Okay, so now let's talk about how k-means clustering works, using the example of pizza places in San Francisco again. So the first step in k-means clustering is to put 3 cluster centers randomly on the graph, which are represented here by the 3 stars. The next step is to find the set of points that are closest to each center and partition them into groups indicated by the 3 colors here. So the algorithm, again, iterates through this process. So first we move the stars to the center of the points in the new labeled groups. And then we calculate the distances again between the center and the stars groups. We calculate we calculate the new set of closest points, and then we move the stars to the center again. And we keep doing this until the algorithm stops moving the stars to the center. So again, how do we know how many groups we have to start with? Because we have to choose. One way this is determined is by repeating this process many times for different numbers of clusters, and then plotting the diameter of each cluster as a function of the number of clusters, which is shown here. And we find that the number of, then after we do this, so we have number of clusters on the x, the diameter of each group on the y-axis, we look to see where the change in cluster diameter begins to plateau. And so you can imagine, for this example, a fourth cluster might split this blue group into two groups here, but that might not be very informative or helpful for our analysis later on. So we want them to stay together. And in general, k-means clustering is good for finding things that have a certain circular geometry, but the shape of gene expression across stable cell physiologies might not be circular or Gaussian. So the world contains more than circles. Okay, so the third, and as of now, most popular and potentially best performing algorithm for cell type clustering of single cell RNA-seq data is graph clustering that uses community detection. And communities are defined as groups of cells with higher probability of being related to each other. So within the group, higher probability of being related to the other cells within the group than being related to cells that are members of other groups. And community detection algorithms find groups of cells where there are more edges inside of the group than edges linking cells to the rest of the graph. So the Leuven and Leuven algorithms are community detection algorithms that optimize for modularity. And modularity is a measure of how densely connected nodes within a module or a cluster are relative to the density of connections between nodes belonging to different modules. So in general, in graph-based clustering approaches, there are two types of graphs that can be built. The first is the k nearest neighbor graph, which we talked about yesterday. And this connects vertices or nodes, in this case cells, based on whether they are among the k number of smallest distances away from each node. So the second is the shared nearest neighbor graph, where the connection between nodes or the edges are weighted by the number of nearest neighbors the cells have in common. So let's look at that a little bit closer. K nearest neighbor graphs are constructed using adjacency graphs, where you have a symmetric matrix. And if two nodes are within the k number of closest nearest nodes to each other, their relationship is marked by a 1. And if they are not close neighbors, they are assigned a 0. So in this example, we consider the 3 nearest neighbors. So the number of k is 3, and we ask which are the 2 closest neighbors to node B. And by measuring the distance, we find that nodes A, C, and D are the closest nodes. So as you can see, these nodes receive a 1 in the adjacency graph, while nodes B, E, and F receive a 0 because they're not the 3 closest neighbors. And to make a weighted matrix, we calculate information about the set of shared neighbors between 2 points. So for point B, the connection to node A is weighted the strongest. It's 0.8 here, followed by C, because they share a similar set of neighbors. In contrast, the relationship between point B, and D is not weighted strongly because other than themselves, they lack a shared set of neighbors. So the Leuven algorithm, or the slightly optimized version of the Leuven algorithm, are the current clustering methods that are recommended as the best practice approach to single-cell RNA-seq data. The Vane algorithm calculates communities based on the shared nearest neighbor graph, where each link between nodes is weighted for how many shared nearest neighbors each connected cell has. And then there are two additional steps in the Leuven algorithm. They're simple. The first step is modularity optimization. And here each node is iteratively moved around into different modules until the density of connection within a module relative to the number of connections between the module is maximized. And then the second step is community aggregation, where each node is labeled with the community it belongs to. And so, k-means clustering employs a set of geometrical assumptions about what cell identity shapes look like. The Leuven algorithm is biased for enriching regions of high density, which may or may not reflect similar physiologies. Okay, so what happens when we apply these three clustering algorithms to the same set of data? Well, as you might expect, we get different results. So then in this experiment, k-means, hierarchical, and Leuven clustering were applied to a synthetic set of data. And the synthetic single-cell RNA-seq data was simulated in such a way where there was one set of distinct cells, a second set. Sorry. The distinct cells have relatively low transcriptional variation and are indicated in red here. The second set has greater variation and is indicated in this top left cloud up here. And then the third set was simulated to have continuous variation in expression along some sort of manifold space, which is indicated in purple here. And then there is a final rare set of cells off in the corner here. And as you can see, these three clustering algorithms give qualitatively different results. K-means clustering fails to distinguish this rare cell type from the trajectory of cells with continuous variation. Hierarchical clustering was able to recover this rare cell group, but it fails to partition this trajectory of cells and also this group of cells with more variation. That's okay. And Leuven was able to partition this trajectory of cells, but the rare population was grouped into one of the groups from the trajectory. So out of many different algorithms and procedures and also hyperparameters, how can we evaluate which set of partitions is the best? So one quantitative way to evaluate cluster similarity is using the adjusted rand index, or the ARI. And essentially what the ARI does is calculate the similarity between two clusters based on overlap in their set of cells. And then we adjust the score to account for the maximum similarity, which might occur at random. So the ARI measurement can be used to compare similarity in cluster membership across different methods. So for example, if we have our one set of data and we perform two different clustering attempts on the data, we can then ask whether or not the same set of cells are in cluster 1 attempt 1 as cluster 1 attempt 2. So if cluster membership is exactly the same, and we have the same set of cells in both clusters, we would get an ARI score of 1. If the clusters are totally different, we would get an ARI of 0. And the stability of ARI scores, as well as the total number of cells within a cluster, can both be used as a measure of clustering stability and potentially be used as a proxy to assess whether some stable mathematical object exists that can be reliably partitioned using our algorithm of choice. And here the ARI scores are also useful when comparing to a set with a priori labeled truth values. So if you do facts beforehand, you can ask whether or not how well your clustering recovers the set of known facts labels. And this is the experiment that was kind of done in this slide. The truth is slightly modified to be a fake truth. I'm not sure why that But so in this slide, I'm illustrating the abundance of different clustering pipelines that are used in the analysis of single-cell RNA-seq data. And one of it's just one of the reasons this field is so challenging to keep up with. So in this publication by Duo et al., the authors calculated the ARI for different single-cell RNA-seq clustering approaches compared to data that had been annotated as the truth. And like I said, it's unfortunately not fully a ground truth, so these scores should be taken with a grain of salt. But anyway, the group then clustered the ARI scores of different clustering algorithms using hierarchical clustering. And what we see is a dendrogram of clustering approaches. And you can see that Seurat is most related to the fake truth, and then to this slightly more computationally intensive SC3 algorithm, which uses a consensus approach to generate self-partitions after subsampling the data many times. But what I like most about this graph is that it illustrates many different combinations of how to cluster cells. So the bottom row is in different shades of green, and they indicate whether the implementation is performed using your raw counts or the log normalized counts or other ways in the distance matrix. And then the second row, the top row in shades of red indicates whether PCA, t-SNE, or no dimensionality reduction technique was applied to the data. And then the middle row in different shades of blue indicate which type of clustering algorithm was implemented. Where is ScanPy? Great question. ScanPy replicates Seurat, so we can lump those together. Okay, so ultimately the takeaway message from this is that clustering is subjective. First of all, no perfect measurement of all state variables of a cell, as well as their future trajectories, exists, so we can't perfectly map partitions in gene expression space to cell physiology. And second, biologists have to make decisions about what's most important to pay attention to, which is often founded in the history of the tradition. And because single-cell RNA-seq is a relatively new technology, the current assumptions and choices that go into clustering pipelines are not super well founded in empirical evidence or theory. And many of these procedures were developed on test data that mixed two very distinct cell lines together, performed sequencing, and then attempted to recover these two very distinct groups. But the shape of biological variation in more complex tissues is not as well understood. So some of the principal choices. As I mentioned yesterday, very briefly in the chat, that the Euclidean distance metrics that quantifies the distance between two cells in gene expression space is potentially problematic because it more heavily weighs genes that have higher expression values, just by virtue of those genes contributing larger numbers to the distance matrix. And this doesn't necessarily sit well with people because whether the magnitude of mRNA abundance maps to differences in cell physiology is not well established. So I can think of numerous examples in metabolism where higher mRNA counts do not map linearly to the performance of a physiological function like glycolysis. Instead, it's more the ratio of different gene expressions that might better indicate flux through certain metabolites. But anyway, the current consensus of the field is to perform clustering on a dimensionality reduced set of features using the Euclidean distance. And that's okay because typically clustering is an iterative process. It's performed, a hypothesis is generated by the clustering results, and then it's experimentally evaluated in a physical system. So these experiments can either support or invalidate the single-cell RNA-seq experiment, and science keeps going on. But before experiments are performed, there do exist some ways to evaluate the quality of your clusters. Yes, instead of absolute abundance, some people do use z-scores of abundance to normalize over all the genes, regardless of the abundance, to weigh them equally. And I had a discussion about this once that I'm blanking on right now, but if you remind me, Harry, I can get back to you. So one thing that's often done is bootstrapping, and bootstrapping implies that you randomly subsample your data or downsample so that you're only considering a fraction of your cells, and then you perform the clustering pipeline over and over again to ask whether you see robust or similar clusters. So then you can use this approach to generate confidence intervals for your quantitative analysis of groups. For example, if you want to measure how the composition of cell types in your tissue changes after a drug, you can randomly subsample a subset of your cells, say 10 different times, and each time you recluster the cells, annotate the cell types again, and calculate the proportion of each cell type in your data. And then you can generate a confidence interval around the mean fraction to demonstrate that your findings are statistically robust. So another important thing to do is to always check the quality control metrics of your clusters. So to do this, you can graph different quality control features on top of each cell when visualizing which cells belong to which cluster. This can reveal whether there are patterns caused by batch differences, read counts, or number of genes per sample. Though some words of caution here, the stemness of a cell has been shown to positively correlate with the number of different genes detected in it. So this is evidence to say that there can be some biological reason for having the number of genes or the number of total read counts correlate with the partitioning of your groups. Okay. And finally, the experimental way to validate clusters is by performing orthogonal measurements of the system you're studying to see whether or not the expression patterns of your clusters and the labels of your clusters match cell type patterns in the system you're studying. So here the authors are doing in situ hybridization to color specific mRNA transcripts that were identified on genes in a single cell RNA-Seq study of Hydra. So to summarize clustering, the goal of clustering is to find subpopulations of cells that vary in similar ways. Some of the challenges inherent in this work is that our notion of fixed cell identities can limit the types of questions we asked about gene expression, and clustering can be performed on many different subsets of our count by cell matrix, often with dimensionality reduction being applied first. But we also know that all dimensionality reduction tools induce distortion on the data structure. So we have to be careful when choosing how and which tools we apply. Also, there are many different types of clustering algorithms that find different kinds of clusters that start with different assumptions about geometric properties of your cell type. However, without a systematic understanding of how cell physiologies map to transcriptional spaces, it's hard to evaluate which clustering pipelines employ the most appropriate set of assumptions about the shape of gene expression variation within populations of cells. So if any of you are about to create some single cell RNA-Seq data, I think it would be super helpful to the field if you could try to do your best to orthogonally annotate cells before sequencing them. There seems to be lacking a lot of studies that link cell physiology to gene expression, in part because it's challenging to do. So, so before we go on to downstream analysis, I'll pause here for questions. And in case you're wondering, again, these cells are the cells that secrete the gel that holds your brain together to stop it from collapsing in on itself. They're called the choroid plexus layer. Okay, I'll carry on then. So after we have our cell clusters and we've mapped them with some notion of cell type, either using previous orthogonal measurements like FACS or looking for marker genes that we know to be relevant to our system and annotating our cell types, a natural question to ask next is which features distinguish these different types of cells, or which features distinguish two experimental conditions. And do these features or genes map to biologically interesting processes? And these kind of studies can try to address the question also of how cell types transition from one identity to another, say in like development or normal tissue homeostasis. So some common frameworks for exploring these types of questions are compositional analysis, or how cell types change in their relative abundance over different experimental conditions. Another is trajectory analysis, which attempts to reconstruct the passage of cells and their expression patterns from one type to the next. And finally, differential gene expression, which simply compares two test sets of cells to look for differentially expressed genes that are different between the groups. Okay, so first let's talk about differential gene expression analysis. The goal of differential gene expression is to simply identify which genes are significantly over or under expressed across different groups or conditions. And to do this, we can use simple statistical approaches to test whether the mean of this gene is different between two test groups. And to do this, people test each gene's mean expression level between two test groups, either using a t-test or a Wilcoxon-Rank-Sum test. And the Wilcoxon-Rank-Sum test is more robust because it corrects for false discovery rates and uses rank data. So another important point is that differential expression is performed on the raw data, as this is the most accurate representation of your gene's expression. So you don't want to use your normalized data here. So after we perform our clustering algorithm and assign cluster indexes to each cell in our data matrix, we go back to using the raw count data for the analysis of quantitative differences in gene expression. And it's important that this is the most accurate representation of our original sample. So one downside of this approach is that these methods are highly sensitive to how we draw the partitions between the groups of cells to begin with, as well as other factors such as read depth or how many genes you find within each group of cells. Though overall, it's a fairly straightforward statistic to calculate. There also, however, exists some concern in the field that because the nature of single-cell RNA-Seq data is that there are very many zeros in the data, and there are potentially distributions that are bimodal in different ways, this comparison of means might not be the most appropriate method to identify differentially expressed genes. So if you look at the literature, there exist several model-based approaches that try to overcome these issues of multimodality and sparseness to improve on the identification of differentially expressed genes. But modeling the data can be difficult, and certain kinds of models also have higher false discovery rates. So in this paper by Mao et al., they tested the reproducibility of methods and their ability to re-identify the same set of genes when the data is resampled. using subsets of the cells. And they found that monocle and edge R were not very reproducible, but all of the methods across the board struggle in identifying differentially expressed genes that have low expression levels. So in another example, differential expression testing also becomes more difficult when attempting to test for differences in means between two different experimental batches. So for example, say you have single-cell RNA-seq data collected from 2 patients, and you'd like to find genes that are differentially expressed across a specific cell type from each of the 2 patients. The systematic differences between the 2 patient samples, if they're prepared independently, can affect the result of a Wilcoxon-Ransom test. So in theory, it would be possible to work around this issue by doing a logistic regression using the patient as the latent variable. And then as a result, you'd get a p-value for how much each gene contributes to the difference between the 2 cell types after controlling for the differences between the patients. However, to do this, you need to make many strong assumptions about the distributions you're working with. And as I've said before, this is very difficult to do in practice because of the diversity of cell types within samples and the unknown distributions of genes within them. To summarize, there's lots of practical issues in trying to regress out batch effects, given the number of genes and it being a more parametric approach. Is that a cat? Yes. Okay, so once we've collected our sets of differentially expressed genes from each cell cluster, they might be pretty long lists. So something that can be useful to do is perform a gene set analysis. So essentially researchers plug in the differential expression results into the Go machine, the gene ontology database. And this assigns physiological processes to your genes. So as a result, you then get some information about which cellular processes are overrepresented in your cell type clusters or across different experimental conditions. And using these Go terms can help you make inferences about how the cell clusters in your data correspond to the physiology of your tissue. So another direction the field is moving in is toward inference of gene regulatory networks. And this approach attempts to infer relationships between genes as a way to predict differences in gene expression dynamics across trajectories of cells. And in single-cell RNA-seq studies, it uses co-expression matrices as the input, and it's highly dependent on pseudotime trajectories. So not a ton of tools have been developed for this, and based on comparison to real-time course data and known regulatory networks, none of the tools perform well. However, this kind of analysis is gaining popularity in multimodal data, where researchers try to map changes, particularly in chromatin accessibility, with changes in gene expression profiles at the single cell level. Okay. So I've mentioned trajectory analysis. So now let's talk about how it works. And the goal of trajectory analysis is to reconstruct the path of cell differentiation using single-cell RNA-seq data. So the idea is to ask questions about how cells transition from one cell fate to another during development. But this is also important for understanding the ongoing process of tissue maintenance, as illustrated here, where enterocytes, the cells that line the surface of your intestine, mature as they traverse out of intestinal crypts. So the question is, it looks like there are many different algorithms which give better or worse results depending on the nature of RNA-seq data. That is true. And is there something like a cheat sheet to choose appropriate algorithms or pipelines based on the data and questions being asked? And kind of. There are a ton of benchmarking studies that try to test how well each of these different kind of methods are doing. And I've read a lot of them. But I think what I find most frustrating when I look at these data is they don't often have like a ground truth data that they're comparing to. They often use simulated data or just unsupervised clustering to ask whether things can recover the unsupervised clusters. So I think there's a real lack of like ground truth data to validate different methods in the field. And it can be hard to navigate. But if you do have a specific question in mind, you can like think carefully about what your question is and the best way to go about answering it. But I'm afraid that's all I can advise. I think there's something like 40 different pseudotime algorithms. There's a chart that I saw, and usually whoever's writing the paper, their method is at the top. It's not suspicious at all. But so in addition to being a tool that can be used to study actively developing or differentiating cells, trajectory analysis can also be applied to cells that are not expected to cluster into discrete cell types. So in other words, you can use trajectory analysis to analyze any set of cells that might be expected to display continuous changes in gene expression. So when analyzing continuous processes, it's common to forego clustering altogether and instead order the cells along a one-dimensional manifold, and this manifold is called pseudotime. Oops. Okay, so in the major assumption of pseudotime is that the developmental landscape of a cell can be modeled by a smooth continuous surface. But as in cell clustering algorithms, where the algorithm's job is to produce clusters, pseudotime's job is to produce an axis of pseudotime. And this may or may not reflect an underlying biological reality. So I think this concept of smooth developmental landscapes has been carried through this century by this illustration of Waddington's landscape, where stem cells slowly roll down this landscape of potential energy and take paths into canals where stem cells develop into their terminal differentiation state. But whether this is an appropriate model for how changes in gene expression map to cell differentiation has not been experimentally tested because it's hard to do. So some issues you can imagine is what if the balls are rolling at different speeds, or what if some cells aren't moving on this landscape at all? Pseudotime only takes the still frame of single cell RNA-Seq data and projects all of the cells, both the developing and non-developing, onto the same one-dimensional manifold. So the bystanders, non-moving cells would be trapped. But also, what if the developmental process is not represented by smooth continuous changes in gene expression? It's also a real possibility, as it's been shown that many genes are oscillatory in their gene expression patterns, and a pseudotime algorithm would take a gene with nonlinear dynamics and attempt to order that cell along a manifold with gradually increasing levels of that gene, which would totally misrepresent the underlying dynamics. So how can we measure how well a pseudotime algorithm works? Again, we need orthogonally annotated data where we know a priori the temporal order of gene expression states. So here, again, we have our data where cells from different stages of early embryo development were sequenced. And so we have the zygote stage 2, 4, 8, cell, and so on. In the ideal pseudotime algorithm, we would be able to line up cells from known temporal stages into a diagonal line where the temporal stages are plotted in pseudotime. So as we can see here, this T-scan algorithm does a pretty decent job at this, though we only have like maybe 200 cells in this sample, unfortunately. But some other algorithms like diffusion maps do not do so well on this data. So again, there are many different kinds of pseudotime algorithms, but they mainly differ in the structure of the graph that's allowed to be built. So here's an illustration of what I mean by this. Some trajectory inference algorithms impose a linear path along data, while some allow for bifurcation and some allow connected graphs or loops, which you can consider to represent the process of transdifferentiation, where one cell type reverts back into a different cell type without following a linear differentiation path. Okay. And so trajectory algorithms essentially work by reconstructing cells so that their changes in gene expression are ordered along the graph. And in this heat map in figure E, you can see that each column represents a cell and each row represents a gene. And so the blocks are colored by the magnitude of the gene's expression. And you can see in this first cluster, the expression of these genes decrease along with pseudotime. And sorry, this cluster. And while many pseudotime algorithms exist, they all mainly suffer from this major issue that assumes linear changes in gene expression patterns over the developmental manifold. So if there exist dynamical patterns in gene expression that are not linearly correlated to each other, such as an increase and a decrease, and then an increase again of different genes, this cannot be recapitulated by pseudotime. So overall, pseudotime represents not necessarily how cells transition between cell types in organisms, but how their transcriptional profiles can be ordered if we line them up according to linear transitions or forced gradients in expression patterns. However, there are other ways. There's different kinds of pseudotime algorithms that take a different approach. And one is called PAGA, which is implemented in ScanPy and shown in figure D here. So instead of forcing cells to line up in some sort of graph, PAGA quantifies the probability that two different clusters are related to each other by estimating the density of shared nearest neighbor edges. So here, in addition to viewing the clusters, we get some information about the probability that different groups of cells are more closely related, which is represented by the thickness of these edges between nodes. So here is just one more example of pseudotime performance on real time course data. So in this experiment, researchers undertook a really large single-cell RNA-Seq analysis project or experiment where they sequenced many different things from the mouse to create kind of an atlas reference project. And one of the sub-experiments they did was take samples from different embryonic stages of the limb development. So within the limb, they, they chopped off the developing arm of the mouse and then separated the tissue into the skin, the immune cells, and the kind of like whatever else was left. And they have samples ranging from embryonic week 10.5 out to 15 as colored by this sunset theme. And the middle row represents the orthogonally annotated labels. So in this case, it's the ground truth, where 15 is the most developed cells, and this blue color is the earlier stages of development. And the top row is the pseudotime axis, and the bottom row is the cell clusters as annotated by the Leuven clustering algorithm. So looking at how this data is projected onto the first two principal components, we can make some observations about how well the pseudotime analysis lines up with the real developmental stage. And you can see how it kind of forces this gradient over time that doesn't really exist in the real data. It's like kind of there, but a lot of the transitions are kind of imposed, and there's different like overlapping spaces on parts of the graph that are totally missed. And the same is kind of true for the clustering. It can identify different bulk dense regions, but it kind of misses the regions that are more in between. So this is a pretty complex looking shape that gets totally missed by the cyan cell type cluster here. So I'm going to pause for questions again before we go and do a final Kahoot quiz and then a demo in ScanPy. So let's take a break. I'll take questions also. And then we'll meet again at 2:50. So there's a question is how reliable is RNA velocity in generating trajectories? And I meant to look into that to refresh my memory before today. I thought you would ask, but I remember we were very shocked at the first version of RNA velocity and some of the assumptions it employed, but the second version tried to improve it a little bit. Let me see. Okay, so the idea of RNA velocity is that it sequences RNAs as they're being transcribed and compares them to the ones that are complete in their transcription. So to interpret the data, we rely on the idea that the expected ratio to splice to unsplice RNA is given by a uniform set of first order parameters. So it's expected to fall in a straight line. If it's lower, RNA goes down in the future, but if it's higher, the RNA will increase in the future. But the concerning thing is that to estimate the shape of this line, they combine all of the data from all of the genes. However, there's orders of magnitude differences, like what was shown in your lab, in the decay rates of different mRNAs. So combining them is problematic for constructing the model. But it's definitely an interesting direction to develop further. Recording. Thank you. All right. Final quiz, except for the assigned one. Here we go. What's the appropriate use of t-SNE and UMAP?
What's the number for the quiz?
Oh, I don't know. Oh, here it is. 8530381. Great. Most of you. For the two that chose cell type clustering, very bad. No. t-SNE and UMAP are mostly used for exploring the data and secondary to like look pretty, so we have some comprehension of how marker genes are distributed through our data and how they overlap with our cell type clusters. They have a completely almost randomized topology at the local and like global level. while it preserves some sort of structure, it's not appropriate to do cell type clustering on this two-dimensional distance space. Okay, true or false? Yes, it's false. They're only designed to preserve pairwise distances, not neighborhood structures of the high dimensional data. Okay, so great. I'm glad most of you got it correct this time. Oh, and I'm sorry, this is an extremely granular image, but I think what I see still a lot in presentations of single-cell RNA-seq data and also in publications is clustering on this UMAP space. I think in general, there's a lot going on in single-cell RNA sequencing and a lot of statistics and different steps, and it's very disorienting. So people have some sort of faith in the biostatisticians and the math that clustering will somehow recover appropriate sets of cells. And also it's pretty, I think, intuitive to look at a t-SNE projection and see patterns in it and make some inferences about how cells move through this t-SNE space. But when we quantify distortion, we can do so by calculating the neighbors in the high dimensional space, like the 20 nearest neighbors of cells, and then the neighbors in the low dimensional space, and comparing how much this set of neighbors overlap relative to the total combined set of the neighbors. So when we look at this overlap of t-SNE versus high dimensional data, we see essentially that 98% of the neighbors are totally different. So it only has its one closest neighbor still. And this is problematic for cell type clustering because you want to find cells that have similar expression patterns across like many dimensions of genes, not just two nonlinear, non-topological distance spaces. So we don't do that. I'm sorry, wait. Next. Okay, so what algorithms can we use to group cells? Yes, we can use whatever clustering algorithms we'd like. However, these either use geometric or density-based assumptions, and you can imagine if gene expression space looked like more of a fractal or a non-convex form, that neither of these assumptions would be appropriate. So unfortunately, we still don't have a lot of information about what's the best thing to do because of the lack of orthogonally labeled data sets. Yes, the ARI is used to compare set similarity and how much overlap two sets would have. In this case, we'd consider cells. So how many cells they share in common compared to how many would be overlapping at random. So how do we do? Awesome, Eve, 5 out of 5. The knowing frog is very wise. The smooth seal for the win. Great job, everyone. Okay, so now let's move on and into our ScanPy tutorial. We will start by looking at how to annotate we'll start by applying clustering algorithms to the PBMC data, and then we will annotate these clusters using marker genes, and then we'll look at differential gene expression across the groups. And then we'll go on to do an experiment to evaluate different clustering pipelines using orthogonally annotated data. So let me share my screen. You're welcome to either, work through this with me or just follow along in this case. And please interrupt me with questions as we go, if you have them. So ScanPy tutorial 3 is the first thing we'll start with, and this is continuous with ScanPy tutorial 2 and 1. It just builds on the quality control and all of the clustering preparation steps like highly variable gene selection and PCA prior to the actual clustering. So I'm not going to repeat these steps with you for now. We can just highlight everything down to the new material. We have to recalculate all our stuff. So again, HVGs, PCA, and then we can make some neighborhood calculations. And lots of figures. Okay. And then here we are. So for today's tutorial, I didn't save all the different annotated data objects as different names, which I asked you to compare yesterday. Here we just have A data. So to make sure all the calculations worked that are there, we can print the name of our annotated data object in the Python console again and check out what happened. So we seem to have greatly reduced the number of genes in our annotated data object by selecting for highly variable genes. We have our 2000 cells, 1800 genes, and then various quality control observations, variables, and then we have our dictionaries where the transformed data lives, as well as the principal component reconstruction scores and distances between cells and also their weighted k-nearest neighbor graphs, so their edges. Okay, so we can first plot, I'm going to close these extra figures. We can plot our data in UMAP coordinates. And then we will use this coordinate set to project our cluster labels after we do the Leuven clustering, which is a slightly modified but not very different version than Leuven. And for all of these functions, if you want to learn more precisely about what the code is doing, there's documentation on the ScanPy website, and you can also find the source code to look precisely about what's happening. But there you can find all the different arguments that you can choose to pass the function. So in this case, if you hover over it just right, it would also tell you, oh, there we go. So there are it's kind of easier to look at it on the web page, but here we're using a resolution of 1. So this hyperparameter, to remind you, controls the coarseness of the clustering. If you have higher values, you have more clusters. If you have lower resolution, you get larger, more granular clusters. And depending on how we change that, we'll change the resolution of our study. So let's see what it looks like. Okay. And then we can plot the clusters on our UMAP coordinates. And we end up with 9 different cell types, which we may or may not expect based on our knowledge of peripheral blood mononuclear cells, which I don't have a lot of. And you'll notice Python starts counting at 0, whereas a lot of other coding languages start with 1, and this can cause some confusion when indexing in Python. And also we have our cluster 0. And we can look at how it corresponds to like known marker genes. And we see here that our CST group has been subdivided to 4 different clusters. There's these kind of like appendage looking things, and this continent is split in half, whereas we have this totally distinct cell type over here that doesn't have any of these known markers. And then this kind of peninsula looking gradient of this marker gene that's kind of bisected into 2 groups. So if you wanted to save the results, you could write the results file, and this will save your labels on your cell. So it'll save the numbers that are generated that assign each cell into its appropriate group. So another way of looking at the relatedness of groups of genes is the Pathograph. So we can easily look at what that looks like and how related our different clusters are to each other by using this built-in ScanPy function, which you can read about more. And plotting, we can color that either by the cluster labels or our marker genes of interest. It plots one graph, it prints one graph for every marker gene you specify as a string in this list of color. So we can see here that it kind of reflects what we see on the UMAP, where these 4 clusters are pretty densely related to each other, with 1 and 0 being more similar and 4 and 6 being more similar, which we might expect if we know that NKG7 is a really important gene marker for the system we're studying. And similarly, there's less chance of these 3 groups being different, because they're all like pretty strongly related, and that kind of reflects what we see with this distribution of CST3. So we can also recompute our UMAP by initializing it with PAGA information. See what that does to our graph. And while it looks very different, it might give a totally different picture of what we think is going on, but also difficult to interpret. No one really knows what UMAP means. Okay, so the next thing in the analysis is you have these clusters, and we have these two genes that we know about, but how do other known marker genes map to our annotated cell clusters? And are these genes also listed within the genes that are ranked as differentially expressed? So we can kind of test our expectations about different cell type expression patterns. And the first thing to do here is to calculate the differentially expressed genes. So sorry, this is in the wrong order. So first we'll compute the ranking for differential genes across each cluster, and then we can plot them against our marker gene. Oh, then we can use this gene list of differential expressed genes to pull out the ones that we know stuff about or are interested in pursuing further based on the body of work that's been done before us, and use these as our marker genes that distinguish different cell classes. Okay. So here we're just computing the rank of the genes across the different groups in our A data groups. It knows how to do this because the annotated data object has the cell cluster indices. We're using the Leuven organized groups. You can also set any key if you were to have orthogonally annotated data. You can add a list to partition your data differently, and you're choosing the method, in this case, Wilcoxon rank-sum test. And then this second graph here is just the plotting modularity that will show us the 25 most differentially expressed genes across our 9 cell type groups. And I don't know who made this decision, but again, we're plotting the names of the genes as the data labels in order of the most significantly differentially expressed to the least. This is just the top 25, and their rank scores. And then we're comparing each group to the cumulative set of the rest of the cell cluster groups. So this is the genes that distinguish group 0. They're mostly like ribosomal proteins. So maybe this group 0 might be more proliferative cell type or metabolically active in other ways. And whereas the rest of these gene groups seem to have some more specific immune cell indications. Okay. So we pulled out some of these reference genes, which we can pass to Python by assigning this list of strings to the variable marker genes. We can read the results file that we generated from this marker gene test and plot the data in a different way. So here we're just plotting it in a table. So the head command just shows the top 5 differentially expressed genes across each group, but also so that you can output this to an Excel file for follow-up studies. And to do that, it's useful to convert your data into a pandas data frame. So this is pulling out the ranked gene groups, making a new frame that's organized by the groups, and adding the gene names to our group. And then using this pandas data frame, we can start to look at different ways of plotting our data. Okay. So there's also other ways to look at differential gene expression it's limited because it can only compare groups of cells. It cannot look at what genes are more heterogeneous within a group, so it misses that kind of variation. But we can either compare specific groups to the whole data set, or we can look at group by group comparison using this reference argument. So say we're interested in group 2 and 3 as potentially related cell types, and we want to know what genes distinguish them. We can perform this test again, but just comparing 2 and 3, and look at what that looks like. But this time we'll do it in a slightly different way. So we'll plot a violin plot. So this is the output of our first rank gene test, and our violin plot. Appears to be missing. Interesting. Oh no, I can't see the chat again. Would you be able to ask the question using your microphone, or should I stop my share?
I ran that code and it threw an error.
Which
I'm just copying. It said key error Leiden.
Oh, so maybe that would indicate that we don't have the data matrix named the Leiden. So here, nope. Here is where we name the groups Leiden. Actually, it's further back. The Leiden function should do that by itself, unless you missed this line.
Line 74?
Yeah.
Huh. Okay.
Hopefully these others violin plots work. Here we go. So this is more visually informative way to look at differentially expressed genes that give us some intuition about how its levels vary across our groups of cells. So here we can see that our CST3 has kind of a range of expression in this group 2, but is more highly concentrated in group 4, 5, and 7, for example. And similar with NKG7, we can see how the density of the gene's expression changes relative to all the cells per group. And we can also modify these graphs in many different ways, like by color or by changing the labels. So people who studied the immune sites, the peripheral blood mononuclear cells, looked at the differentially expressed genes and inferred that they most likely corresponded to these established kind of classical discrete cell categories of the immune system. So we can pass new cluster names to our annotated data object and rename the categories of Leiden, which right now are just 0 to 8 with these cluster names, to make our plotting a little bit more intuitive. When we say like plot group 2 instead, we can say plot T cells. And we can also plot those labels directly on this UMAP that appears to be lacking axes. And there's a lot of different ways to visualize these differential expression patterns. You can use heat maps, you can use these kernel density estimators, which are the violin plots, or box plot if you prefer. There's a whole tutorial on the ScanPy website of different ways of data visualization for differential gene expression. Here I'll just show you two more before we go on to a more experimental kind of tutorial. I think this one is one of my favorites because you can look at many different genes and across all of your cell clusters and start to notice potential patterns in different groups of genes that are expressed. So here we're just plotting our marker genes, but you can do this for all of your differentially expressed genes. You can have a lot of different outputs. You can also potentially, instead of color the groups by their clusters, you can color them in a similar way as a dot plot, where the intensity of the color reflects the average count for that gene. Here, it tries to like simplify this these violin plots just into a single dot, where the size and intensity represents the intensity of each gene. So here's this one gene that I know what it is. GNLY is a protease that helps break down bacterial cell membranes. So as you can see, we find this in I would expect to see it in natural killer cells, but we don't actually, and that's kind of strange. So maybe our clustering is a little bit weird. Interesting. Okay, so here I have a lot of instructions about how you can save your results to CSV files or compress your files to a zip format to save disk space. But overall, I think I presented a lot of commands to you, but if you go through this slowly and follow along with your data or the ScanPy website, it's pretty easy to use as long as you have some informed understanding about what each of the functions do to your data and how you might want to modify some of the inputs, like, for example, adjusting the clustering resolution parameter. If you get way more clusters than you expect to see, you could dial it down. Or more quantitatively, if you get a lot of clusters that are pretty similar, you can also reduce the number of partitions. Does anyone have any more questions on the ScanPy tutorial before I move on? Okay. So to clear out my workspace, I'm just going to exit the Python console to remove all those variables from the memory. And then re-import my libraries and go through line by line this experiment that asks how could we compare different clustering pipelines. So in this experiment, you might want to load this data while I explain it. So the question here is, how well do cell clusters calculated from different feature sets compare to cell types identified with FACS? And you can do this experiment with any permutation of the pre-processing and pre-clustering steps to evaluate how stable your clustering results are. And in this particular experiment, we have the added information of facts labels. So in the publication by Duo et al., they identified different types of immune cells by before they sequenced the cells using fluorescent activated cell sorting. So they use antibodies to bind to specific cell surface markers that correspond to known immune physiologies. And these labels are retained as the name for each cell in this cell by gene matrix that I just loaded as a data file. Okay, so in this experiment, feature selection was computed using two different approaches. So first, I generated highly variable genes using the generalized linear model that segments the most variable genes out of subsets, the most variable genes, those genes that have the highest coefficient of variation relative to their mean expression levels. And then I also used the feature selection model proposed in the Townsend et al. Paper that I sent, which uses a binomial model of capture, which basically says if we know the genes, if we have the average gene expression level for each gene, we can develop a capture model where we have an expectation of how many cells we would expect to observe the gene in. And if we observe the gene in fewer cells than expected based on the average level, then we can infer that that gene is not expressed in a subset of our cells, our data, so that gene is differentially distributed, meaning it's only represented in a subset of cells. And in that way, we think it might be biologically variable. So it's a way of measuring a difference in the distribution rather than a higher level of variance. So using these two feature sets, we can then perform Leuven clustering on three different count by cell matrices. We will look at just the raw UMI counts, and then also the two feature selected sets, the HVG by cell matrix and the DDG by cell matrix. And we'll take a look at how different the clusters are by visualizing the data using t-SNE, and then we can quantify the difference to see which procedure was able to best recover the original facts labels. And do this quantification using the adjusted RAND index. Okay. So in the interest of time, I'm going to kind of walk through the pre-processing a little bit, just the clustering for the first raw data set, and then I've pre-computed some of the other cluster indices for this visualization. All right, so we've loaded our data by using the ScanPy read. We can also read in, instead of sparse matrix formats, we can also just pass it normal CSV files, like an Excel spreadsheet with our count by cell data. And then we can also load in the facts labels, which are different annotated barcode and index information sheet. So we have the cell IDs, and we can call this index the facts clustering in this new PD data frame called that's designed to designate each cell index list from the three different clustering experiments. Okay. For our plotting input, this list of commands simply replaces this list of strings with a list of numbers so that we can plot things according to their number value, because that's what we need to do. So now we're going to perform the Vane clustering on just the raw count by cell data. So what does our count by cell data look like again? Let's just remind ourselves by typing raw data. So now we have this object, and it does in fact have almost 16,000 different cells, and there's 16,000 different genes and 4,000 different cells. So here there are 500 cells from each cell group represented. And we will calculate our neighborhood graph on the non-PCA reduced data by passing it the userep_x command, which is not super intuitive, but it's detailed in this documentation about how to do that. And we will calculate the neighborhood graph using 20 nearest neighbors. And then we'll compute our community detection with the Leuven algorithm in a resolution of 0.5 across all the data sets, and add this key of all genes to our ScanPy object. And once we do that, we can take the labels out and put them into our pandas data frame where we're collecting all the different cluster indices. Okay. So after we do the clustering on the data, we can visualize what our clusters look like using t-SNE. However, this time it's a bit more exciting because we can compare how our cluster labels look like compared to the FACS labels that we obtained before we did the sequencing and that we have for each individual cell. So here I will plot two graphs. One will be colored by the Leuven clustering, and the other will be colored by the FACS labels. And manifold approximation algorithms like t-SNE and UMAP can take a while. All right, so here's our results, our first set of results out of 3. So when we do Leuven clustering, okay, so first let's look at the facts. We see here that there are 3 distinct groups that kind of fall into their own region on this t-SNE projection. And these are the real ground truth labels. So there's distinct antibodies that correspond to, this is B cells and monocytes and natural killer cells. And then there's kind of this T cell clusters. There's regulatory T cells and helper T cells and cytotoxic T cells and progenitors of each of those. And as you can see, they're kind of all like intermeshed in this t-SNE projection where we can't really distinguish them very well from each other. There's kind of like 2 lobes here. When we do Leuven clustering to identify regions of densely connected cells using all of the genes, we do an okay job. We can recover the 3 distinct groups, but there's obviously some trouble separating these more intermeshed groups of cells. So now let's see whether feature selection can help us. And if you wanted to, you can also color these t-SNE plots by the concentration of a specific gene. So I'm going to do that just now to show you this is kind of like the appropriate use of t-SNE. It's just like, oh, look at that. The CD52 gene is kind of expressed in a lot of the immune cells, which can be expected. And this AIF1 gene belongs particularly to this cell cluster. Okay, so to speed up the demo, I already calculated the clustering and t-SNE coordinates on the HVG and DGG data. It's essentially just this same step repeated, but instead of inputting the raw data, I passed the argument that corresponded to those specific feature selected data. And here we are just using the raw counts, so I didn't include any of the normalization when doing the clustering. However, normalization was used to select the highly variable genes, but it's not used for the identifying these differentially distributed genes. Okay, so now I'm just going, I save, and from the annotated data object, you can save the t-SNE coordinates as they're a separate observation into like a CSV file for easier, faster loading if you wanted to replot it later on. Okay, so we can load those files in, and we can do some formatting to change the row labels to an index, which is a pandas format Python. We can then create data frames just for easy plotting with their cluster labels versus their facts labels for these two data sets. And then here I just manually curated the clusters so that we can more easily compare them across the three different distance matrices. Okay, so now let's plot our t-SNE graphs first with the clusters computed using the HVGs. And then we can plot them again with our categorical facts labels. And again, this uses the HVG as the HVG data matrix as the basis. And what do we get? So here is the truth data, and here are the Leuven clusters. So when we use the highly variable genes as a basis, which enriches for those genes that have the most high variances relative to the mean, we see that we kind of lose some sort of neighborhood structure maybe in these overlapping groups of T cells, especially when we compare this to what the raw data graphs look like. So you can see here, there's kind of more shapes here that are not as easily distinguished in the HVD data. However, when we do the Leuven clustering, it seems to like kind of, but not really correspond to the same set of, to the appropriate cell labels, where we're kind of picking up on this green group and maybe most of these pink groups, but these three cell types over here are maybe not well partitioned by Vane Algorithm applied to HVGs. So now let's see whether this different feature selection model does anything. And what you might first notice is this looks a little bit more like what the raw data looks like, where the two lobes are kind of separated, but they're still kind of meshed. And we do see maybe a better separation potentially by the DDGs. There's a little bit more structure anyway, but it's hard to really say. So what can we do? We can choose a more quantitative method to compare how well these different feature selection methods do at recovering the original facts labels. So to do that, we will use the adjusted RAND index, And this compares set overlap, set membership between our Leuven labels and the original facts labels. So we can do that across all three of our test groups. So we have just the raw counts as clustered by Leuven, HVGs and DDGs, and we'll compare all three to the facts and plot the result. Does anyone want to bet which one wins? All right, let's see. Okay, so the winner is Leuven clustering on the DGGs. The HVGs present a small improvement over the all genes, but also they're all kind of similar and they all kind of don't do a great job at distinguishing those different T cell groups. But overall, we can see how different clustering procedures will generate different results and tell us which genes are differentially expressed will be different based on what we use. So having some a priori knowledge can kind of help us inform what decisions we want to make. Like maybe a different approach to this analysis would not to use so many partitions in this group, since it seems to do a not so great job. Like maybe instead we just want to consider this subset of T cells, which are related T cell lineage, as just one group for our analysis. I don't know. So, sorry, this one's more of a following along experiment. Does anyone have any questions about Python before I kind of wrap up the workshop? Okay, hopefully some of these tools will help you if you plan to do some of your own single-cell RNA-Seq experiments. And if you don't, hopefully you feel a little bit better prepared about how to interpret some of the literature that's out there. Bye. Okay, so I just have a couple slides, reminders to tell you before we wrap up. Completely. Okay, so to recap, we've gone through the entire single-cell RNA sequencing analysis or data acquisition and analysis pipeline. And ideally, you have a better understanding of what clustering can and cannot do in recovering specific cell types from your data, and how you might go about identifying different kinds of cell types and attempting to interpret this extremely high-dimensional data. The goals of this workshop were to introduce you to all of the single-cell RNA sequencing concepts, familiarize you with the experimental approaches, as well as analytical techniques. And hopefully you found this informative or interesting. So in conclusion, to wrap up, single-cell RNA sequencing, the analysis of the data is very involved. There's several different steps, and the goals overlap with the other steps and in the process, and how all of this comes together to affect the results is not super well understood. We know that the raw data collected is susceptible to noise and distortion along multiple levels of the process, first in the data acquisition, but also in the dimensionality reduction when we tried to correct for this noise. We want to avoid overcorrecting as much as we can. But current methodologies, while well-meaning to approach specific challenges, they can be informed by vague or arbitrary or historical concepts and allow bias to enter the experiment. So generally, I recommend using models that have the least strongest assumptions in the analysis procedure and that seem to be reasonable to you and other people who you know who maybe have more expertise in the area. Like method improvement is a very active area of research, and there's lots of online resources available that can help you navigate what method is right for you, including websites like Towards Data Science, Geeks for Geeks, and things like that. But also, I'll be here and you can reach out to the collaboratory. There's also a number of other very talented postdocs who are available for further questions, and as well as collaborations and consultations if you're doing single-cell RNA-Seq analysis projects. We will be here for further help along the way. And you can help us get better by filling out a survey about how this workshop went. So your feedback would be super important for me, and you can help me adjust this workshop to better meet people's needs in the future. And also, do not forget, for those who are taking this class for credit, there is a quiz in the Google Drive folder, as well as a homework assignment that should be due next week, but I can be flexible on. So here, I'll just leave this comic here. So hopefully we can share some laughs in the struggle. And thank you everyone for listening and for asking great questions along the way. Also, if you have any more questions, there's some extra time and I can help you with Python.
So the quiz is also in the workshop or in the Google Drive, sorry.
Yeah, I'll send out an email to people who need to take it. Anyone's welcome to take it to just test their knowledge. But it's in the kind of the parent folder. So in class materials, there's a quiz.
Okay.
Not in day 1, day 2, or day 3.
Okay, perfect, thanks.
Also send out details for the homework assignment, which is just going to be writing a short news and views style article of one of 2 papers of your choosing.